Hey folks, and welcome to another edition of Zog Science. Today we're going to be taking our final look at Section 8. We'll be looking at responses to the environment from both animals, plants, and a little bit of single-celled organisms as well. So just kind of in general, how life works in a very basic, very broken down sense, is that the environment has some sort of stimulus, which is going to affect the organism. The organism is then going to respond to the environment, which can sometimes change the environment, and then that is going to then have a new stimulus on the organism, and it is a continuous cycle. So, for example, a mole is foraging. If there is no food, then the mole is going to move on. However, if there is food, the mole is going to stay there and bite on the mole, right, and eat it. And then eventually, once the food is absent, the mole will continue on. And this is, again, just kind of a basic way of kind of how life works and how systems, how organisms deal with the environment. So let's take a look at some different responses to some different stimuli. The first one is phototropism, and that is a growth in response to a light source. So photolight tropism, and it is mediated by auxin in plants. We've talked about auxin before when we talked about different plant hormones. And so what happens is that the illuminated side of the coleoptile, or really any part of the plant, is going to experience less growth than the, not less growth, but less elongation than the shaded side of the plant. And what we can see is that that response is actually coming from the tip. So when the tip is removed, you can see that the plant itself does not grow to the side. If the tip is covered with a cap, it also does not grow to the side. However, if the tip is covered by a transparent cap, the light can reach the tip and it grows over to the side. Or if the actual site of the curvature were to be covered, then it would still be able to grow over to the side. So something is going on in the tip of the plant, and that is where the auxin is actually being produced. And so what they did was they did some other different experiments where they put the auxin on different kind of sides of the plant, and you can see that there were some different responses. And basically, this is where they kind of figured out that auxin was the thing that actually had the control. They would separate the tip using a permeable membrane versus an impermeable membrane, and the tip grew to the side when it was permeable, not when it was impermeable. So the tip is where the auxin is actually being produced. How it actually works is that what is known as the acid growth mechanism. So the auxin increases proton pump activity, and that is going to pump more hydrogen ions into the cell wall, so from the cell into the cell wall. And those increases in the hydrogen ions in the cell wall activate expansins, and those expansins cause the cell wall to elongate, as you can see here. And what happens is that because this is happening all along the shaded side, all of those cells elongate, and that causes the plant to tip over towards sunlight, towards whatever other light there is. So that's one way the light can be affecting a plant growth and have it lead to some sort of response. Another is photoperiodism. This is where we can have changes in the plant behavior due to the length of day or night. In plants, it is controlled by phytochromes. So phytochromes are these cool proteins, and they have a kinase at the bottom that can have some sort of activity, and then they have actual photoreceptors up at the top, and they control kind of which wavelengths of light have more or less effect. That should be an E, not an A. Have more or less effect in any particular circumstance. So, for example, in general, there are some phytochromes that deal with more blue light phototropism. So they tend to be only affected by blue lights, where there are other responses that are more due to red lights, which we will talk about here in just a moment. So in this case, the phytochrome that we're looking at is PFR-PR. And the PFR form is induced by red light. So the exposure to red light leads to the conversion from the PR form into the PFR form. And that PFR form leads to various responses. For example, seed germination, control of flowering, some other things. Now, sunlight contains both the red and the far red. However, the conversion of PR into PFR takes less time than PFR turning into PR. Therefore, you will accumulate PFR faster, and that is actually how seeds will germinate. And the reason why they always tell you when you're planting seeds, you don't want to plant them too far underneath the soil because they actually need exposure to red light in order to germinate. So the PFR will accumulate inside the seed, and once it gets to a certain level, that is when seed germination will occur. Now, plants also don't want to make sure that they are in sunlight, right? Because you want to have photosynthesis occurring. So if they are in the shade, that is going to filter out red light, which is going to leave more far red light, which is going to induce a greater amount of PR. And that PR is going to limit lateral growth and induce vertical growth. And that's going to help allow the plant to grow up until it gets into the sunlight. Once it is in the sunlight, it's going to want to spread out as much as possible to get as much light for photosynthesis as it can. And so once it gets up there, it is going to be creating more PFR. Once it has more PFR, that limits vertical growth and promotes lateral growth. Another type of conversion process that can occur is that PFR overnight will change into PR, and that can have various effects on the plants and help it to kind of get help it to affect its circadian rhythm. There are some plants that are short plants, short day plants, and other plants that are long day plants. And basically what this means is that they flower when there are particular environmental conditions in terms of the days and nights. So a short day plant will flower when the days are short, so when the days are less than usually around 14 hours or so. And then a long day plant is only going to flower when the days are longer than 14 hours. And so therefore they will not be flowering, when either their pollinator isn't around or, when other conditions aren't right. So some flowers only flower in the winter, right? But that would be a short day, whereas some flowers only flower in the summer. That would be a long day. And you can affect that mechanism by shining light on the plants. So by flashing light during particular, during the darkness, you can affect whether or not they will flower or not. And that's because you're going to be resetting that PR-PFR ratio throughout the night. And again, you can just use far red or near red spectrums, and you can impact whether or not short day plants or long day plants are able to actually flower or not. And one of the kind of interesting things about this is that it's not actually the daylight that matters. So if you were to turn off, the dark, make it dark for a moment and then turn it back to being light, you wouldn't actually impact whether or not the flowers would flower or not. It's all about what we call the critical dark period. So basically, is it dark for a long enough period of time or not? It's not actually whether or not there is light, it's the absence of light. So pretty interesting stuff. There has also been some experimental evidence that shows that there is a protein called FloraJun that is produced by leaves exposed to favorable flowering conditions, so whether or not you're a short day or a long day. And so that protein is basically believed to be the same between all plants, because if you take a long day plant and you graft it onto a short day plant and you expose it to short day conditions, it will actually flower as the short day plant is flowering, even though it is a long day plant. And so it is believed that protein will function between any plants. And another interesting thing about that protein is that if you were to just shine light on one of the leaves of the plant and it was a long day plant, it would still flower even though the rest of it had been in darkness for too long. So I think that's kind of neat and interesting and weird as well, that this protein is produced by the leaves that then induces flowering. All right, circadian rhythms. I'm sure you've heard of them before. These are 24-hour cycles that are mediated by environmental cues that then have an impact on physiology, for example, melatonin levels or bean plant leaves. So in some plants, they have a circadian rhythm where they go through different changes in their physiology, whether they're at night or at day. And you can see that here. The bean plant has put its leaves down even though it's in the light while it's midnight, because that is its 24-hour cycle. Now, the cycles can oscillate a little bit and change if they are taken out of the natural environment. So bringing a plant inside, it will actually oscillate a little bit. It won't be on exactly a 24-hour cycle. It might be something like a 27-hour cycle, or maybe like a 20-hour cycle. But if you keep them out in the light, they will maintain a rigid 24-hour cycle. For humans, our circadian rhythm is generally impacted by melatonin levels, and we have different things that are we tend to be able to do at different times of night. So late at night is when our melatonin secretion starts. That is around 9 o'clock, which is when the sun tends to start going down. And that is part of the reason why you get tired at night, is because your melatonin levels have decreased. I'm sorry, your melatonin levels have increased. You stop secreting melatonin in the morning. You tend to wake up around that time, and then you're able to do all these kinds of different things because your melatonin levels are low. And as your melatonin levels run out, you have some physiological changes, and then you start to secrete melatonin again, and that again impacts different parts of your body. So pretty cool stuff. Now, when you experience jet lag, which is something that if you've ever traveled across country or gone other places, this is where you desynchronize your internal clock. And what happens is that your internal clock is releasing melatonin on a certain schedule, but if you're getting different environmental cues, so for example, your body's telling you that it's 6 a.m. based on melatonin levels, but it's really 3 o'clock in the morning, you might wake up because your body's like, hey, I'm releasing this, I'm starting to produce melatonin. I'm sorry, I'm not producing melatonin anymore. It's time to wake up. Well, it's not light outside. So that's the process of jet lag is you have a different environmental cue from what your internal signal is, and so that messes you up. It's kind of like motion sickness, where your eyes are seeing something different than what your inner ear is feeling. In terms of it messing up the external signals with what's going on internally in your body. Hibernation and migration are some other examples of things that are regulated by changes in physiology and day length. There are some organisms that are obligate hibernators, where if you were to put them into a 24-hour light cycle, they would still hibernate at some point, even if they had the exact same day-night times, they would still hibernate. But in general, hibernation is kind of triggered by shortening of the days. Also, temperature changes can have some sort of impact there as well. So what they did with some squirrels was that they measured their body temperatures and metabolisms over time. And what they found was that their body temperatures would kind of dip down, and then they would kind of spike every once in a while during the period of migration. And those spikes were correlated with their rates of metabolic activity, and they were basically, keeping their burrow a little bit warmer than the outside temperature. However, overall, their body temperature was, was suppressed because they weren't using as much energy, which is kind of the whole point of hibernation. Taxis and kinesis are negative feedback loops in response to various environmental conditions. These are all movements, however, they are slightly different. I'm not terribly concerned that you know the exact specific differences between the taxis and kinesis, but I think in general you should be able to understand the difference. A taxis is where you have an oriented movement towards or away from a stimulus. So this is a very directed movement with a direction. If you've been physics, think about a vector quantity that has a specific direction rather than a scalar quantity that does not have a particular direction. This would be kind of a good way to think about this. A kinesis is a non-directional response to a stimulus. So the organism is being stimulated, it's going to change, but it's not going in a particular direction. However, it is looking for something else than what it currently has. So a good example of a kinesis, it would be a sow bug. It's going to move much more quickly until it finds a moist site. So this is, sow bug is kind of another name for a pill bug, which we've been doing experiments with. But it's moving around. They move around a lot, and once they find a moist site, they chill out. Right? It's not necessarily looking for that moist site. It's not a directional thing, but once it finds it, it knows where it wants to be. And then we've got a taxis. This is where you've got, let's say, phototaxis be moving towards light. In this case, the trout is orienting itself in the face to face the direction of the current. Why? Well, there's going to be more likely to be food coming down the river current, so it's going to orient itself to be able to face in that direction. So again, taxis, think about it like a vector, right? It's got a specific direction. Kinesis, more like a scalar quantity, doesn't necessarily have a particular direction. There are a lot of different types of taxis. You can have phototaxis, chemotaxis, phonotaxis. You can have geotaxis, magnetotaxis. There's a lot of different types of taxis out there. It just totally depends upon what the stimulus is and kind of what the situation is. Quorum sensing. We've talked about this briefly before. Quorum sensing is a way of communicating and cooperating among single-celled organisms. And allows them to respond to their environment, which is all the organisms around them. And basically what happens is that you have individuals that, when there's a low cell density, they're producing these chemical signals that can act as transcription factors. And when there aren't very many of them, those transcription factors are not at a high enough concentration to actually affect a change in the genetic transcription of the organisms. However, as cell density increases, you have a much higher concentration of the transcription factors that is going to lead to changes in gene expression. And in the example of cuttlefish have bacteria in their actual arms that will, at high enough densities, they will begin to fluoresce. And the cuttlefish can actually control their density by pulling in and excreting water, which is basically means that the cuttlefish can control whether or not they are glowing or not, which is a pretty cool thing. But quorum sensing is a fairly new discovery. It's something that's been discovered within the last 5 or 10 years, and it's really just kind of being explored a lot. It's one of the reasons why people think that there's kind of a lag time between you initially getting a disease like a bacteria, some sort of illness, and then you actually experiencing symptoms, is that the bacteria have to build up enough of a concentration to be able to secrete enough of the transcription factors to actually begin to have a pathogenic effect, because they don't want to jump on you immediately. They want to make sure there's enough of them to then be able to kind of attack, loosely as that term can be applied here, which is I think is really interesting and has a lot of implications for medicine. Because what if we could stop those signals? What if we could identify what those molecules are that are being produced and produce drugs that shut those down? So pretty interesting stuff. Another chemical signal that animals use, not single-celled organisms, are called pheromones. These are chemical signals between different multicellular organisms, and they're extensively involved in communication. Insects use pheromones a lot in terms of communicating. Bees, will be attracted to pheromones in flowers. Plants produce pheromones in order to either attract predatory insects that can eat insects that are trying to eat them. They use pheromones to attract pollinators. So a lot of interesting chemical things going on there. Ants will lay down pheromone trails to lead from food to their nests, so that way they don't get lost. Pheromones are produced by us. You and I are producing pheromones. Those are generally involved in mammals. They're generally involved in some sort of sexual attraction. So if you've ever heard of like a dog being in heat, that is when the female is ovulating and she's actually releasing a pheromone, and that is helping to communicate to male dogs that, hey, I'm available for reproduction at this time. I'm available for mating. So pheromones used are in those types of things. That's basically what their goal is to facilitate communication between different animals. We can also have visual communication. So rather than because we have the ability to sense to sense light and different visual patterns, visual displays are used. Again, these are usually associated with some sort of mating behavior because you're needing to communicate between two organisms. Basically, you're saying, "Hey, look at me! I'm fit for mating," things like that. And these are found in most sexually reproducing species. There's all kinds of different visual displays that are used, and it's all about being able to communicate. So that's all I've got for you today. This is the end of Unit 8, which is organismal physiology. So once we come back from spring break, we'll be getting into ecology. Thanks for watching, and I'll see you next time.